Something 26 feet long and weighing close to 12,000 pounds is standing its ground in what is now Montana, three horns forward, a massive bony frill shielding its neck, facing down the single most dangerous predator on the continent. Triceratops horridus was not a passive animal. Its two brow horns alone could stretch past 3 feet in length, mounted on a skull that could measure nearly 7 feet from frill to beak, one of the largest skulls of any land animal in Earth's history, land or sea, living or extinct. Fossilized bite marks discovered on Triceratops bones, matched precisely to the tooth spacing of Tyrannosaurus rex, remains some of the best physical evidence anywhere of exactly how that predator actually hunted, and several of those wounds show signs of healing, meaning the Triceratops survived the encounter and lived on afterward. Then, 66 million years ago, the same asteroid that ended Tyrannosaurus rex ended Triceratops too, along with every other non-avian dinosaur on Earth, in a single geological instant that closed out more than 150 million years of dinosaur history. What it left behind was bone and horn scattered across the same rock formation as its old rival, and in one specific case, in a fragment of horn pulled out of the ground in 2012, something that should never have survived at all, wrapped inside a controversy that had almost nothing to do with the science itself. The obstacle that defines Triceratops research at the molecular level is identical to the one facing every other dinosaur: the DNA itself. Ancient genetic material has a hard ceiling on preservation, degrading beyond recovery after roughly 1 to 1.5 million years under even the most favorable cold, dry, chemically stable conditions. Triceratops went extinct 66 million years ago, a gap more than 40 times beyond that outer limit. Whatever genetic code this animal carried is gone permanently in that form, and no method available to science can bring it back. Claims of recoverable dinosaur DNA belong to the same category of fiction that once suggested amber-trapped insects could preserve a dinosaur's genome intact. Those claims were never independently replicated and are now understood as contamination, not evidence. Named in 1889 by paleontologist Othniel Charles Marsh, Triceratops has been studied for well over a century, and for nearly all of that time, the idea that anything organic could still be sitting inside its bones was never seriously entertained. But in May of 2012, a team excavating in the Hell Creek Formation of Montana pulled a massive Triceratops supraorbital horn out of the ground, and what was eventually found inside it reopened a question paleontology thought Tyrannosaurus rex had already settled 7 years earlier: not whether DNA could survive, which it cannot, but whether something else, something sturdier, still could. The excavation was led by Mark Armitage, at the time the manager of a microscopy laboratory at California State University, Northridge, working alongside Dr. Kevin Anderson, a microbiologist from Arkansas State University. The horn they recovered was one of the largest ever pulled from that site, more than 3 feet long, and the team paid the landowner $3,000 for the fossil before hauling it back to the lab in California. Once there, standard laboratory processing was used to remove the fossil's mineral content and examine whatever organic material might remain underneath. What came out of that horn, once the mineral content was gone, was material that, by every rule of fossilization chemistry, should not have still existed at all. If you are finding this interesting, be sure to subscribe to understand more DNA from the past. Under microscopic examination, Armitage and Anderson documented soft tissue structures consistent with osteocytes, the cells responsible for maintaining living bone, along with thin branching extensions typical of that cell type. None of it showed any sign of mineralization or crystallization, meaning the material had not fossilized in the traditional sense at all. It was the first report of this kind of soft tissue recovered from Triceratops horn material anywhere in the scientific literature, extending a small but growing body of similar findings that had previously come only from species like Tyrannosaurus rex and a duck-billed hadrosaur, and adding an entirely new category of dinosaur specimen, horn material specifically, to the list of tissue types known to preserve this way. The two researchers submitted their findings to Acta Histochemica, a peer-reviewed journal specializing in cell and tissue research, and the paper was accepted and published in February of 2013 under the title Soft Sheets of Fibrillar Bone from a fossil of the supraorbital horn of the dinosaur Triceratops horridus. The paper itself made no claims whatsoever about the age of the fossil. It was a technical, descriptive account of unusual cellular structures, nothing more. 2 weeks after publication, Armitage was terminated from his position at the university. The school stated the decision was the result of a departmental restructuring unrelated to his research, a routine administrative explanation that universities give for staffing changes on a regular basis. Armitage disputed that explanation and filed a lawsuit against the university the following year, alleging religious discrimination. Armitage is a board member of the Creation Research Society, an organization that holds that the Earth is a few thousand years old, and according to court filings, he had discussed with students his personal view that the preserved, non-mineralized condition of the tissue suggested the horn could not actually be 66 million years old, a position he later described publicly as, in his words, the obvious conclusion once you had seen the tissue firsthand. That personal interpretation was never part of the published paper itself, and it is not a position shared by the broader scientific community studying dinosaur soft tissue. A faculty member learned of those conversations and reported them, and Armitage's contract was not renewed shortly afterward. The university fought the lawsuit for roughly 2 years, arguing the termination was purely administrative before a judge declined to dismiss the case outright and allowed it to proceed toward trial. In 2016, California State University, Northridge, settled with Armitage for $399,500, bringing the 2-year legal fight to a close before it reached a jury trial. Armitage's attorney called the outcome precedent-setting, noting he was not aware of another case where a creationist plaintiff had received a comparable settlement in a dispute of this kind. CSUN, for its part, was explicit that the settlement resolved an employment claim and did not constitute any concession on the merits of the underlying science, and the university maintained the original termination had been budgetary rather than retaliatory. A settlement ends a legal dispute about whether someone was fired for an unlawful reason. It does not and cannot adjudicate the age of a fossil or the validity of radiometric dating. That distinction matters because the actual scientific question the tissue raised, how can soft, unmineralized cellular material survive inside bone for tens of millions of years, already had an answer by the time this controversy played out, and it did not require revising the age of the fossil record by even 1 year. In 2005, Mary Schweitzer's team had discovered similar soft tissue inside a Tyrannosaurus rex femur, triggering more than a decade of skepticism, replication, and testing across multiple independent labs, before the finding was broadly accepted as genuine rather than contamination. By 2013, that same year, Schweitzer's team had identified the mechanism. Iron released from breaking down hemoglobin at the moment of death drives a chemical process called the Fenton reaction, cross-linking surrounding proteins into an extremely stable structure, essentially preserving the tissue chemically from the inside out. The same way a lab fixative like formaldehyde works, except triggered automatically by the animal's own blood chemistry with no human involvement at all. Laboratory testing showed this effect could increase tissue stability more than 200-fold, extending survival at room temperature from a matter of days to more than 2 years under controlled conditions. Scaled across geological time inside a sealed, mineral-encased fossil, that mechanism provides a fully evidence-based explanation for exactly the kind of preservation Armitage and Anderson had photographed in the Triceratops horn, using ordinary biochemistry operating within the standard well-established timescale that dating methods across multiple independent techniques continue to confirm. Subscribe to not miss the next one. The finding also did not remain an isolated data point. In May of 2023, Armitage returned to the exact same collection site in the Hell Creek Formation, this time working with researcher Jonas Cruz to examine surface-level bone fragments that had never been excavated or previously studied. Using the same laboratory process, the team again recovered soft osteocytes, structures consistent with blood vessels, and what appeared to be peripheral nerve filaments. Their results were presented at a meeting of the Microscopy Society of America and formally published in 2024 in the journal Microscopy and Microanalysis. The follow-up findings reinforced that the original 2013 discovery had not been a one-time fluke tied to a single, uniquely preserved horn. Whatever process was preserving this material at that specific site appeared to be operating consistently across multiple separate fragments collected more than a decade apart. Triceratops soft tissue is now one entry in a small but expanding catalog of confirmed non-avian dinosaur specimens, showing this kind of cellular level preservation, joining Tyrannosaurus rex and at least one hadrosaur species in the peer-reviewed literature. Each of these findings has followed a similar arc: an unexpected discovery, waves of justified scientific skepticism, independent replication using separate techniques and separate research teams, and eventually a chemical explanation grounded entirely in known biochemistry rather than any need to reconsider deep time itself. Radiometric dating of the volcanic ash layers within the Hell Creek Formation, cross-checked using multiple independent decay systems including uranium-lead and argon-argon dating, continues to place these fossils at approximately 66 to 68 million years old, entirely consistent with the broader geological record and the well-documented timing of the asteroid impact that ended the age of dinosaurs. Nothing about the preserved tissue itself, examined on its own chemical terms and compared against the mechanism Schweitzer's team identified, requires the timeline to be wrong. What actually happened here is two separate stories that got permanently tangled together in public memory, each one frequently cited without the other. The first is a legitimate, peer-reviewed scientific discovery. Original, non-fossilized cellular material recovered from a Triceratops horn, adding a third dinosaur species to a growing list of confirmed soft tissue finds, later reinforced by an independent follow-up study conducted at the same site more than a decade afterward, using the same methods on entirely different fragments. The second is an employment dispute, resolved through a six-figure settlement that addressed workplace discrimination law specifically, not paleontology, geology, or the age of the Earth, and settled on terms that explicitly left the underlying scientific questions untouched. The tissue itself does not know or care what conclusions anyone drew from it after the fact. It exists because of iron, not ideology, sealed inside a horn that once belonged to an animal that spent its life standing three horns forward against the most dangerous predator its world had ever produced, and lost that fight only when the entire sky changed at once, 66 million years before any of this was ever dug back out of the ground. Decades before a microscope and an acid bath in a California lab would finally show anyone what had been quietly sitting inside it the entire time, waiting for the right chemistry and the right question to finally reveal it.